Comprehensive molecular characterization of papillary renal carcinoma by Andrea Pinzo de Dispa, Medical English, Dr. John Moscoso. Definition, application in medicine, pros and cons, issue applied in Ecuador, conclusion. Definition of papillary renal cell carcinoma. Papillary renal cell carcinoma is a type of cancer that occurs in the kidneys. It accounts for about 20 and 50% of all renal cell carcinomas. Renal cell carcinomas are a type of kidney cancer that develop in the lining of very small tubes in the kidney. PRCC can be divided into two types. Type 1, which is more common and usually grows more slowly, and type 2, which are usually more aggressive.
So interesting in the context of kidney cancer is that we have 7 FDA-approved drugs for the disease, but we actually don't apply any of these targeted drugs in a specific fashion based on a patient's genetic profile. So with the Foundation Medicine Company, I've actually been working with the group to profile a series of over 440 patients with advanced kidney cancer. I've done this in association with my colleagues at the Dana-Farber and at MD Anderson. So what we've yielded from this project is a very comprehensive analysis of this patient population and found multiple mutations that were previously undescribed in the disease. So with this project, I think we'll move one step towards actually identifying the right drug for the right patient. It could potentially lead to major breakthroughs. And I think that's going to be a big paradigm shift in the way that we do clinical trials in the future for this disease. So, in the context of this project, amongst these 443 patients, we actually have a smattering of individuals with rare subtypes of kidney cancer. Papillary kidney cancer is certainly one of them. At the U.S. Congress of Pathologists, we actually reported some data in that subset. We actually are reporting out data at this meeting pertaining to collecting duct kidney cancer. And this represents just about 1% of all patients with advanced kidney cancer, but it happens to represent the most aggressive subtype of the disease. It's invariably fatal. It responds poorly to chemotherapy. The chemotherapy represents the standard. What we've uncovered in collecting duct carcinoma are mutations, several that may potentially predispose to certain targeted therapies. So for instance, we've uncovered mutations in a gene called NF2. This may potentially lead to sensitivity to drugs like everolimus or Afinitor. So I can really see a continuum with this research that leads to targeted therapies for these rare subtypes of the disease.
Applications in medicine. Papillary renal cell carcinoma is the second most common subtype of RCC. Two subtypes of PRCC are recognized. Type 1, in which the papillae are lined by a single layer of cells with scant pale cytoplasm, and type 2, in which the papillae are lined by pseudostratific cuboidal to columnar epithelial cells with abundant eosinophilic cytoplasms and prominent eosinophilic nucleoli.
With a central fibrovascular course, it is not uncommon to see other types of growth. We have an example of glomerular structures that resemble normal renal corpuscles. Here we have a small area of solid growth, and here we have an example of tubular or acinar type of growth. with a few foci that slightly resembles small glomerular structures. Third useful sign of papillary renal cell carcinoma is the focal accumulation of these foamy macrophages. So here, right papillary carcinoma. Thanks for watching.
Pros. The medication listed below have been approved by the Food and Drug Administration as orphan products for treatment of this condition, sorafenib and aldesleukin.
This was actually the largest papillary RCC to date. We took over 100 patients with papillary RCC, gave them a drug called savolitinib. Savolitinib is a specific MET-only inhibitor, MET tyrosine kinase inhibitor, and has a safety profile that is pretty well acceptable. And why a MET inhibitor in papillary RCC? Now there is data. There is data from several sources, including the TCGA, that there's a subgroup of patients with papillary RCC, this rare really tumor. This is just a small subset of renal cell cancer. All what we talked about renal cell and the trial with IO drug are in clear cell. But papillary RCC is the second most common, still very rare, and we don't understand the biology. But again, a few years ago now, we understand a subset of them do harbor alteration in the MET gene. MET mutation, MET amplification, and others, maybe around 30 to 40%, 50%, and these patients are sensitive to MET inhibitor. There was prior studies and there's a preclinical evidence. So we treated all of the patients with foritinib with metastatic, previously treated or untreated patient with papillary RCC. Now the response rate was low in the total cohort, 8%, but the responders were, the only responders were the ones that had MET alteration. If you had MET alteration, response rate close to 20%, if you don't, 0%. The progression-free survival, totally significant. The tumor shrinkage, also significant if you have MET alteration. And so hopefully in the future, we will have a study, or at least we interrogate this drug that is well tolerated, doesn't have, the VEGF inhibitors toxicity, but in a population that have MET alterations.
Cons. Kidney cancer or renal cell carcinoma is not a single disease, but is made up of various types of cancer that are characteristic by different genetic drivers. Each type has distinct histologic features and distinct clinical course and response to therapy. It's a heterogeneous disease with histological subtypes and variation in both disease progression and patient outcome.
So this was
Issue applied in Ecuador. Age presentation range from early adulthood to old age, with peak incidence in the 6 and 7 decades, mean 50 and 57 years, similar to other RCC subtypes. Conclusion: Type 1 and Type 2 papillary renal cell carcinomas were shown to be clinically and biologically distinct. Alteration in the MET pathway were associated with type 1, and activation of NRF2R pathway was associated with type 2. CDKN2A loss and CIMP in type 2 conveyed a poor prognosis. Furthermore, type 2 papillary renal cell carcinoma consists of at least 3 subtypes based on molecular and phenotypic features.